Hello, ladies and gentlemen. Thank you all for joining us from different parts of the world. My name is Rashmi Rajendra. I will be your moderator for today's event. I would like to introduce our speaker for today. Dr. Katya Nikolov is a physician scientist with over 10 years of academic and industrial experience in cancer biology and cell imaging. Dr. Nikolov obtained her MD from Russian National Research Medical University, and did her postdoctoral research at Memorial Sloan Kettering Cancer Center before moving to the industry. Her background has been in solid tumor models, tumor microenvironment, and immunotherapy, and she has led the development of 3D microtumor assays from biopsies to predict patients' response to therapy and to improve outcomes. Her experience also includes T-cell-based platform for drug screens in senescence research. Here at Protein Fluidics, Dr. Nikolov leads the projects on all 3D cell-based assay development and assay optimization. Her work involves innovating 3D cell-based applications with the PUMA system, in-depth, high-content imaging, and metabolomic analysis. So, go ahead, Katya.
Thank you, Rashmi, for an introduction. Can you see and hear me well?
Yes.
Great. Well, thank you everyone for joining us today. I'm really happy to talk to you about our end-to-end 3D cell culture and assay automation and present our new development, new product, PUMA system with environmental control. But we'll start first talking about the context of our work and why scientists these days turn more and more to 3D cell models. I will, of course, introduce you to our new product, PUMA system with environmental control, and explain how it works. I'll share with you our complete workflow from cells to data, which we developed, and I'll show you how we use this workflow using the application example, and phenotypic profiling of epithelial and mesenchymal markers in breast cancer models. But first, of course, I'd like to acknowledge our partners and collaborators without whom this work won't be possible. I'd like to thank our partners, Yokogawa team, for their support and for their great technology, Cell Voyager's CQ1 spinning disk system, which we use to acquire our beautiful images. Dr. Burrell Group at Tulane University, who kindly provided to us patient-derived triple-negative breast cancer models that they have developed in their lab. And Greiner Bayer team, we implemented their nanoshuttle cell magnetization technology and use it to support assembly of 3D cell models and assay automation in our flowchip. So why there's such an interest in 3D cell culture models? The simple answer is because we need to better predict human biology and acquire clinically useful information. There are many factors that affect our body function, our genetic background, what we eat, our metabolism, the environment we live in, the drugs we take. And as a result, we can develop different conditions, pathological conditions, which will be associated with specific biomarkers that can be measured, symptoms, responses to treatment, and different disease outcomes. And so ideally, we would like to have a model, in vitro model, that would simulate these tissue-specific physiological and pathophysiological environments and responses, and would tell us of what we would see in real human body under similar conditions. And then using modern computational tools and the data that we obtain from these models, we then can draw a more complete picture of disease progression, again, responses to different drugs, towards the goal to improve drug discovery process and to provide fast and reliable information to clinicians so that they can use it to select the best and the most appropriate strategy treatment for their patients. Of course, I cannot not mention here huge news, which is passage of FDA Modernization Act. So for the first time in the history, drug developers have the opportunity to forgo animal models in preclinical development And this opens the door for accelerated adoption of different clinically relevant physiological ex vivo tumor models, again, with the goal to improve predictability and translatability from assay and well to patients. And of course, this initiative will now stimulate even more the development of sophisticated preclinical models and assay systems and use them for preclinical studies. So traditionally, two-dimensional cell culture have been used for decades, and it is still a dominant method in many biological studies. However, we know that growing cells in a monolayer is fundamentally different from how normal and cancer cells exist in human biology. Our body is not a flat layer of cells, like a piece of paper. Our biology is much more complex. And therefore, of course, our goal is to create preclinical models for understanding disease progression and how we respond to different treatments, which would reflect that complexity. From spheroids, talking about 3D cell-based models, from spheroids to organoids and organ-on-a-chip technologies, 3D cell models lie in a spectrum of its complexity and physiological relevance. And such evolution in these models and research, and as 3D cell cultures rise in popularity, they bring unique challenges. 3D cell culture methods still require solid expertise in handling and optimization to build good models and assays. And then utilizing 3D cultures with standard cell analysis methods, which were originally developed to support 2D culture, is not that trivial and can be quite challenging. And so therefore, at Protein Fluidics, we have a great team who puts lots of effort into development of our microfluidic Puma system, the technology that helps scientists to overcome these difficulties and to support them in all variety of their 3D cell-based applications. So what exactly Puma platform brings to your lab? Well, when we work with 2D cell culture, the processes are pretty straightforward. Cells are attached to the surface and relatively easy to manipulate. But once we move to 3D cell culture, things change. They either can be suspended in media, or they can be embedded in matrix, which possess certain difficulties. Often we hear from scientists that they lose up to 65% of their sample during the assay manipulation and run. They damage their structures. If you'd like to run multiple assays using your samples, often you have to prepare a stack of plates. It is not that easy to add and remove reagents, especially if you do some assays that requires multiple steps like that. Often, if you need to do imaging, you have to take your sample out of the plate and either do cumbersome paraffin embedding or transfer it to the glass slide for the imaging, which is also not that very convenient. And the last but not least, of course, we can use expensive liquid handling system, but again, they're kind of pricey, and often they require special expertise, excuse me, to handle them and operate. And so Puma system really comes to solve many of these difficulties. First of all, our protected sample chamber technology prevents a sample from damage, loss, or drying during assay run and manipulation and for any other stresses. This technology enables sequential fluid transfer, so you can really automate complex multi-step assays. You can run several assays at the same time to get more data. And importantly, our flowchip allows imaging inside directly. So you really don't have to take any more sample out, again, go through these difficult processes of embedding, and you can acquire really nice images inside the flowchip. And also the dimensions of the flowchip allows really easy to find your sample during the imaging. And again, important factor, we keep this system affordable to labs. Our pricing is very progressive, and we have different programs so that scientists can get it into the lab. So to summarize, Puma system really allows you to streamline the process of 3D tumor assembly, perform drug or cell therapy treatments, run dynamic multiparametric assays, and at the end, get high quality data. So here I'd like to highlight briefly main features of Puma system. Like I mentioned already, our flow chip is designed so that your 3D sample is cultured inside the protected sample chamber. So sample is constantly covered with the fluid, and such design protects your sample from damage or loss during the assay. In Puma system, you can program automated media exchangers and drug deliveries and set it on a schedule. And in a similar way, you can program automated collection of supernatants that can be further used for the downstream analysis for secreted factors. And finally, it enables automated workflow solution for complex assays like immunofluorescence staining, viability staining, apoptotic assays, drug treatments, and others that you use with your samples. And so today I'm really happy to introduce you to our new instrument, new development in Puma family, Puma system with environmental control. This is a 2-in-1 benchtop incubation and assay automation instrument. The system has to be connected to carbon dioxide source, and it allows you, it controls relative humidity, CO2 concentration, and temperature, and you can initiate the settings from the touch screen. So the system really provides flexibility in a way that if, for example, you'd like to start with cell culture, drug treatments, or any other treatments of the samples, you can initiate cell culture conditions. And then, for example, if you'd like at the end of your assay fix cells and proceed with the staining, you can disable incubator settings and proceed with the assay at room temperature. So you really don't need to bring this system in and out from incubator on your bench or move your sample in and out, everything happening in the same machine. This graphics shows you here how the stability of carbon dioxide and temperature. So after initiation, it reaches preset temperature and CO2 concentration within approximately 5 minutes, and then maintains it stable over the course of your experiment. The core technology, the core part of the system is the flow chip. Here you can see the holder with 4 flow chips in them, and for your experiments you can use between 1 and 4, which allows you to run assays for 8 to 32 samples per run. In this flow chip you can see reservoirs with blue humidity inserts. So this is a new development which allows you to fill up these reservoirs with sterile water and to control humidity during your assay. Each flow chip has 8 lanes, and each single lane is designated to one sample. In this lane, we have central sample well, where you load the organoid or spheroid, and then the central well is connected to reagent wells through microfluidic channels. The spacing of the wells and the frame resembles standard 384 well plates. So this, the whole setup of the plate is compatible with any instrument like plate reader or microscope that you have in your lab. Flow chip can be filled with any reagents like media, compound, staining, stain solutions, depending on your assay configuration. Your sample is loaded into the sample well, and again, it's located at the bottom in the protected sample chamber. This allows reagent to be directed in and out of the sample well without disturbing and drying of your sample. The bottom of the flow chip is a thin cyclic olefin copolymer, which makes it compatible with high-resolution imaging. The volume of the well is small, 20 microliters, so it really saves your reagents when you run these assays. The diameter of the protected sample chamber is 1.2 millimeters, so you can use samples up to 1 millimeter in size for your experiments. This video shows you here how you can load your 3D cell sample to the flow chip, how it is positioned inside the protected sample chamber. And you can see the channels that connect reagent wells with the sample well allow fluid exchange by transferring liquid in and out, again, without disturbing or providing any stresses to your sample. Once your flow chip is loaded with sample and reagents, all you need to do is to select the protocol from a preloaded menu on the machine, press play button and walk away, because all the rest steps will be executed automatically. So now let's take a closer look what is happening inside the flow chip during the assay. Here you can see an example of one line with the sample. We can first remove old media to waste, and then transfer fresh media, set the sample to an incubation. Then we can again drain it out and transfer medium with the compound. Treat your sample with compound for specific amount of time, collect supernatant that can be used for the downstream secreted factor analysis, transfer staining solution, let sample stain, remove the stain, transfer wash buffer, and stop. So at this point, your assay is completed and the plate is ready to be transferred to the microscope or plate reader, depending on what you're doing. So here I'd like to show you two images of MCF7 breast cancer spheroids that were cultured in two different systems. On the left side, this spheroid was cultured in Puma system that was located in the standard cell culture incubator. And then on the right side, spheroids were cultured inside Puma system EC under cell culture environment. And I hope you can appreciate here that these samples are pretty viable. They're happy, nothing happens to them. And this, again, highlights that our EC system provides the necessary environment for culturing spheroids and to ensure that they're viable and happy throughout the experiment. And so now I'd like to share with you a complete workflow that we have developed. We call it From Cells to Data. This workflow starts with seeding cell suspension to the flow chip for 3D cell model generation. So here we use magnetic bioprinting method with nanoshuttle magnetic beads. It helps us to facilitate consistency and reproducibility of the process of 3D structure formation. The way it works is that magnetized cells are loaded to the flow chip on a special magnetic loading tray to enhance cell aggregation, and you can get reproducible, consistent structures formed. Then the culture formation process is supported by automated media exchanges, and as a result, you get nice, viable 3D structure formed in the flow chip, which then can be treated with the drugs or fixed, stained, and imaged directly in the flow chip. So the benefit of this method is that it eliminates the need to transfer your sample from elsewhere for assays, and also at the end, you again don't need to remove your sample from the flow chip and transfer to the glass slide or any other method for imaging. You can do it directly inside the flow chip. So the benefit of this workflow is that all steps are automated, again, without the need to touch your sample during the whole process. And so in this part of the webinar, I'd like to share with you some data that we acquired using these workflows, and I'll show you how we do phenotypic profiling of epithelial, mesenchymal, and proliferation markers in breast cancer 3D models. So for that, we use our developed workflow for immunofluorescent staining. So to characterize the expression of different cellular matrix. And in this protocol, once you're done with treatment of your samples and culturing it, you can fix it directly in the flow chip. And then all you need to do is just load staining reagents in the flow chip, which usually takes you about 15 minutes. And then all the staining steps will be executed automatically by Puma system. So essentially what will happen for your blocking, for antibody incubation and wash buffers, the system will transfer reagents to and from the sample and then incubate depending on the preset time which you select. And after that, the plate with the flow chips is ready to be transferred to the imaging and to acquire your images. So again, the advantage of this workflow is that you really don't need to spend hours by pipetting in and out, and it's a difficult process, especially if you work with structures that are floating in the well. It reduces plastic consumption and it saves you lots of time. And most importantly, it provides pretty good high quality data. So in this experiment, we use two different breast cancer models. We started with MCF7 breast cancer spheroids. These are estrogen and progesterone receptor positive cells. They considered to be low aggressive and non-invasive cell line. And then we also use TUBCX4IC cells provided by Dr. Burlev from Tulane University. These cells were derived from a patient who demonstrated quite aggressive tumor and with resistance to therapy and high metastatic behavior. So here I show you images, maximum intensity projection images that we obtained in this experiment. This MCF7 spheroids and 4AC tumoroids were analyzed for a set of markers expression. We stained them for proliferative K67 marker, and then for ecadherin, keratins, vimentin, and F-actin. And what I show here, first of all, is that MCF7, epithelial MCF7 cells show really strong signal for ecadherin expression and keratins with significantly downregulated expression of vimentin. Whereas on the other hand, patient-derived model with aggressive behavior, 4AC tumoroids, demonstrated very different pattern of expression. We see strong vimentin expression in this model and loss of epithelial markers. So this data highlights that likely these 4IC cells, they display rather mesenchymal phenotype, which is very well in line with the aggressive behavior and highly metastatic nature of the original tumor. And on this slide, I'd like to highlight again the quality of images that you can get. Here you can see 4 images with different markers. These are slices that were selected from a Z-stack. And I hope you can really appreciate the high quality of images that you can get, and you can process them for the downstream image analysis and retrieve lots of data in terms of cell expression intensities and spatial data as well. And so to summarize, our new PUMA system with environmental control enables 3D cell model culture and assay automation. This complete automated workflow for formation, media exchanges, and immunofluorescence staining eliminated difficult sample transfer steps, disturbance, and manual handling errors. Automated immunofluorescence staining combined with high content imaging provides you really good, consistent, and reproducible high-quality, comprehensive phenotypic data. And of course, this platform with all these capabilities can be quite valuable tool in a wide range of applications, including disease modeling, drug discovery, and personalized medicine. So I'd like to share with you also that we have lots of interesting application notes published on the website that are available for download. You can find there lots of useful and interesting information. If you attend or plan to attend AACR in Florida this year, please come see us at our booth and at our poster presentation. We'll be happy to talk to you and learn more about your experiments. And I'd like to thank you today for joining us for this webinar, and I would be happy to take questions.
Thank you, Katya. So a couple of questions for you to begin with. Is the PUMA system compatible with any kind of ECM, gels, matrigel?
Yes, you can use extracellular matrix. Generally speaking, system is agnostic to what type of matrix you use. We've tested matrigel, we've tested engineered hydrogel, vitrogel in our collaboration with the Well Bioscience team. So as long as the gel is compatible with microfluidic, so it might require some optimization, but generally speaking, yes, you can use it.
So, and what type of microscopes or microscopy are the flowchips compatible with?
You can do bright field microscopy, you can do confocal microscopy that we do a lot, of course, with the 3D cell models, because you want to really go deep inside and see these features in the middle of the sphere. So different kinds of systems, whatever you have in your lab. We used EVOS microscopes, we use citation systems, Zeiss microscopes like Yokogawa CQ1, so different kinds of machines.
And can you just clarify about the difference between PUMA system and PUMA system EC in terms of how you do the experiments, etc.?
Yeah, so first of all, if you look at the instrument, it looks pretty much the same. So the difference is that like predecessor of PUMA system EC has to be positioned inside the cell culture incubator if you run your assays under standard cell culture conditions, live samples. And then if you need to do some, for example, immunofluorescence staining and you need to change the conditions, you can take it out and use it on a bench. So PUMA system EC allows you to avoid this. It's a, again, incubator and assay automation in one. So if you would like to culture your system, do media exchanges, do drug treatment, and do live culture, you just initiate environmental settings. So you set your CO2 to 5%, you set temperature to 37, and then there's relative humidity control using these reservoirs with fluid in the flame. And then if you don't need any more environmental control conditions, you can just switch them off, and then system will cool off to room temperature, and you can proceed with other assays.
And have you also tested any kind of, so I mean, I work with you and I know you have working on the immuno-oncology. Could you also tell us a little bit about the immuno-oncology applications? Have you worked on this with the Puma System EC?
These are very interesting applications. So what we've tested is we developed co-culture platform when you have your 3D cell model in the flowchip, and then you can deliver immune cells, for example, T cells or other cell populations to your sample, and then look how these different cell populations interact with each other, do imaging also, you know, to understand to get spatial data. So yeah, these are very interesting applications that can be used for immuno-oncology.
So Simon is asking a question that, do you plan introduction of oxygen control to Puma System?
Yes, it's in the development, but yes.
Also, I can add one more point to you, Simon, that the Puma System EC, even without the development of the oxygen introduction, can be placed into, for example, a Baker-Ruskin chamber, and you can regulate the amount of the environment that it is in, including the Puma System, the original one. And this can control even the oxygen content that it receives within that Baker-Ruskin chamber, for example. So this is something that you can do even at the moment. Katya, one other question that I have for you is, what are the new applications or interesting applications that you are working on at the moment with the EC system?
Oh, there are different kinds of. Of course, we again would like to expand our co-culture applications and to not only limit them to immune cells, but also to introduce maybe fibroblasts and understand in different setups how we can use our system, drug treatments, different kinds of organoids. So yeah, lots
Awesome.
of things. Cell painting. That is very interesting to us, so that's in our plan to develop also.
So if there are no more further questions, I would like to say thank you so much to Katya.
Thank you.